Okay, guys, so we are marching on and continuing in our hierarchy. We're moving upward. So we began talking about the subatomic particles. We talked about what is the atom. We talked about what is a molecule. And then we talked about a micromolecule and then a macromolecule. We talked about the different types of macromolecules. We talked about how we harvest energy. We break down macromolecules into micromolecules, and that is catabolism. Okay, that's exergonic. The energy comes out of the bond. Some people still have problem with endergonic versus exergonic. Endergonic and exergonic are describing, is a convention that describes what's happening in the bond. If there is energy stored in the bond, that's endergonic. Inside the bond, endergonic. I have stored energy in the bond. If I break the bond, if the bond is broken, then the energy leaves, exits the bond. That's exergonic. That's exergonic. So when I catabolize, when I break down macromolecules into micromolecules, the energy exits the bond and I use it. I'm running the marathon. I'm breaking down my fats, breaking down my lipids, breaking down my proteins, breaking down my carbohydrates, then breaking down the energy in the bond. The bond has no more energy. The energy exits the bond. That's exergonic, and that is known as minus delta G. The energy left the bond. But then if I eat pancake, if I have a big meal and I'm laying on the couch, then I'm not burning, I don't need energy, so then I store these micromolecules into macromolecules. So the energy is stored in the bond, it leaves me, enters the bond, and that is positive delta G, endergonic. So that is metabolism. Building up, that is endergonic, that is building up, storing, anabolism. But breaking down, that is known as catabolism, breaking down, as well as no, we're not losing energy, we're gaining energy. We're breaking down the bond, the energy exits to me, so I'm gaining energy. So the problem is you have to identify where is the energy. If I have it, the bond doesn't have it. If the bond has it, I don't have it. We cannot both have energy. Me and the macromolecule. This is the bond within the macromolecule. So if there's energy in the bond, I don't need it. It's stored. If there's no energy in the bond, it exited to supply me with ATP so I can perform some sort of function. So that is macromolecules. Then we realize that these macromolecules and the micromolecules, they're not just there for energy, to store energy, catabolism, anabolism, and both of these are obviously metabolism. Metabolism is the sum of reactions. Some people think that the sum, when you ask them the question, what is the sum of all the biochemical reactions, they say that's homeostasis. No, that's not homeostasis. That's metabolism. Breaking down, storing, breaking down, supplying energy, and storing, building up and storing energy. So that is metabolism. Then we realize that these micromolecules and the macromolecules, they're not just there for energy. And when I say energy, please don't forget, energy storing and energy breakdown. It goes both ways. So ATP is not just there to give you energy. It's there to give you energy and also to store energy. It's like a hanger that you hang your clothes on. So if you have a lot of energy, it's gonna start storing it. It's gonna go from nucleoside to nucleoside AMP, so from nucleoside into nucleoside monophosphate, nucleoside diphosphate, nucleoside triphosphate. It's gonna go upward. So it's nitrogenous base with a ribose sugar, and if you have energy, you're gonna store it as 1 AMP, more energy, 2 di phosphate, then triphosphate. That's when you want to use it as storage. You're storing in the closet. But if you want to break down, then you break down this bond. It goes from ATP to AMP, from ADP to from ATP to ADP, from ADP to AMP, and from AMP to nucleoside. So it goes both ways. Don't think of it as one way. Then we realize that we don't just have our micro and macromolecules for energy. We also have them for structural support of the cell. So the cell has a plasma membrane, and this plasma membrane has phospholipid bilayer. It has proteins embedded, peripheral proteins on the outer leaflet that bind ligands. You have peripheral proteins on the lower leaflet that work like in a signal transduction cascade, this pathway that mediates an effect of an extracellular ligand that cannot make it inside the cell because it is polar and the membrane is blocking it. It's non-polar. And then we know that now there are proteins within the cell that are transmembrane, integral proteins that open up and allow for the passage of ions. Some ions are transported down concentration gradient, others are transported up concentration gradient. So now we have moved up. So now the last thing we want to talk about is genetic expression. How does the cell regulate all this metabolism? How does the cell regulate anabolism and catabolism? How does the cell sustain life? So if somebody who's running a marathon or expending energy is different from somebody laying in the couch. So the processes happening in their cells are different. And they are different because the cell has two capacities. The first capacity is genetic expression. When we say genetic expression, that we mean we turn on and we turn off the process of genetic expression. Genetic expression can mean cell division. So when do we need to divide the cell? Cell division and cell quiescence. So when do we need to divide? Like when there is an injury, we need to heal, we need to regenerate, then we turn on cell division. After we have healed, or also in growth, in embryological development, when the baby is growing up, cell division is active, it's on. But you're gonna reach a level when healing has stopped. So in cell division, that's important for healing. That's after injury or regeneration of tissue, healing and regeneration of tissue. And also cell division is important for growth and development. So if you are going through the stages of infancy to becoming a toddler to becoming an adolescent and then early adulthood and so on, this kind of growth requires a lot of cell division. So we turn that on. But once we satisfy healing and regeneration, and when we satisfy growth, we reach that terminal stage, then we have to turn cell division off. We have to be able to turn it off. So the cell's capacity to turn genetic suppression on and off, that is what enables the cell to regulate the growth and development and the healing through cell division. Okay? Also, the cell can turn on and off enzymes. So there are enzymes that when they are turned on, for example, by phosphorylation, we talked about protein kinases, by phosphorylation. Phosphorylation is when you add a plus pi, when you add a phosphate group to an enzyme. So you see some enzyme is off, is inactive. When you add a plus pi to it, a phosphate group, it will be turned on. But some enzymes, the other way around, they are on. When you add a phosphate group, they become off. So you don't have to worry about that. What you have to worry about is there are mechanisms to regulate the activity of the enzyme. One of them is the protein kinase, the protein kinase system. So the protein kinases, they add a phosphate group, depending on the enzyme. Some enzymes, if you link and you add to them a phosphate group, they will be turned on. And there is a separate set of enzymes that if you add a phosphate group to them, they will get turned off. So it's very interesting. But what's the same thing you need to know is that it's these kinases. These kinases, what is it? You need to know what is a kinase. A kinase is a protein enzyme, and I don't need to say protein enzyme. You already know that a kinase is an enzyme. So it is a protein. So an enzyme is a protein. Not every single enzyme out there is a protein, but pretty much all of them, most of them. So a kinase is a protein enzyme that adds phosphate group. Will this phosphate group activate an enzyme? Will this phosphate group deactivate the enzyme? That's a whole separate story. Now we're gonna go later on to decipher all the different types of pathways, and we're gonna learn about them. But for now, all you need to know is what is a kinase. That's an enzyme that catalyzes the addition of a phosphate group to another enzyme. And the concomitant addition of that phosphate group to that enzyme is going to have different effect depending on the enzyme. Is it going to turn it on or turn it off? Does that make sense? Are we clear? This point is very important. Yes?
Can you repeat the second definition you just gave, that catalyzes? Can you repeat?
Okay, so an enzyme, every single enzyme is a biocatalyst. An enzyme is a biocatalyst. It catalyzes biological reactions. Does that mean every single enzyme catalyzes biological reactions? No, there are physical reactions. There's chemical reactions, but here we're dealing with biochemical reactions like glycolysis, for breaking down of sugars to get ATP and energy out of them, like protein catabolism, like nucleic acid metabolism, all of these, and beta oxidation in fat catabolism, all of these are biological reactions. So when you see an enzyme, immediately say catalyze. It catalyzes. We talked about how the enzyme catalyzes a reaction. And you need to know this graph, and you need to know it really well, and that the x-axis is always going to represent for me the time of the reaction going on, and this represents for me the concentration. And we said that if you want to go from reactants, into products. I'm sorry, this is not concentration, this is energy. I apologize. The y-axis represents the Gibbs free energy, the delta energy, the delta G. So what happens is, in order to go from reactants into products, you need to surmount an activation energy barrier. We need to go up a mountain. And you have two types of energies here. The first type of energy is the energy from the reactant to the product, and this is known as the net free energy. This is the net free energy. And then you have the second type of energy, and that's the energy from the reactant up to the transition state complex. Transition state complex. This is the peak. This is the peak point. That's the saddle. That's the saddle point. That's the top point. So this is EA in purple, activation energy. And this is the net free energy. It's very important for you to understand that the net free energy is never affected. It is the free energy, the purple one. So what does the enzyme do? The enzyme will lower the activation energy barrier. So it will replace a alternative route for the reaction to go. When we drop down the activation energy barrier, the enzyme will say we no longer have to go up a mountain, just go up this hill, B. Go up this orange hill, path B. Don't go through the path A. What happened? The activation energy decreased. So the enzyme decreases the activation energy, speeding up, catalyzing the biochemical reaction. That's why we call them biocatalysts. Did it touch the net free energy? It did not. So the question is, sometimes on the exam when I ask the students, the enzyme catalyzes the reaction by decreasing the net free energy between reactions and products. And they say, oh yes, they click on that. And that is a big wrong. It does not touch the net free energy. That's the first property. It never affects the net free energy. And the enzyme never alters the reaction. So it's never going to give you a different product. If you had AB plus C, and you were meant to have, in the end, AC plus B, the enzyme is not going to give you AD plus CB. It's not going to give you something else, like an Uber ride. It's not going to say, I want to come to this place. It's going to take you, no, it'll take you somewhere else. So it's going to give you the same product, just faster. And it does not get consumed in the reaction. Does not get consumed in the reaction. Does not get consumed in the reaction. It's always there as long as you give it ATP, as long as, this is very important, it needs ATP, needs chemical energy in the form of ATP. It will not take energy in the form of electricity, will never take energy in the form of kinetic energy, it will never take any form, it has to be ATP. To transfer the energy of the bond, of the phosphate bonds, to transfer them into the enzyme and utilize that energy. So we lower our activation energy, but we ensure that the net free energy is not changed. So these kinases, your question was, your name is? Candace. Your question is, what do kinases do? Kinases are, so whenever is a biocatalyst, is the same definition. This biocatalyst increases the rate of the reaction, speeding it up by adding a phosphate group to another enzyme. Now because I added this phosphate group, I have one of two fates. Remember, I have one of two fates. Adding a phosphate group can turn the enzyme on. And adding a phosphate group can turn it off. It depends on the enzyme. But for now, you don't need to worry about that. All you need to know is that what is a kinase? It adds a phosphate. And we talked about kinases when we were dealing with the phospholipid bilayer. We said that the phospholipid bilayer has hydrophilic heads, and it has hydrophobic tails. It has hydrophilic heads and has hydrophobic tails. These are the hydrophilic heads. And these are the hydrophobic tails. Okay, and we said that who's welcome to go through the bilipid membrane leaflet? It is the steroid hormone. What are examples of our steroid hormones that we mentioned? Yeah, excellent. So they are called cholesterol-based hormones. So cholesterol itself is not a hormone, but it is the dough that all the other hormones are made up of. So these, they can penetrate so easily. This is so important. The cholesterol steroid-based hormones are nonpolar. They go inside the nucleus, and they bind transcription factors and they transcribe for us mRNA that will exit out, and then it will get translated in the ribosome to give me my primary structure of the desired protein. And that desired protein that I transcribed because of the steroid-based hormone, and I translate it in the ribosome, that desired protein will give me the target effect. The target effect. I want a target effect. I want to decrease metabolism, increase anabolism. I want to increase catabolism, decrease anabolism. That is my target effect. I want to increase temperature. I want to decrease temperature. I want to increase glucose uptake. I want to decrease glucose uptake. That's my target effect. The target effect is going to be mediated by these hormones. So that is if you have the same identity, likes dissolve like, the same identity of these bilayer leaflet, this non-polar substance. But if you are polar, then you are, nevertheless, you are a first messenger. As long as you're a hormone, whether you are steroid-based hormone, whether you are non-steroid-based hormone, you're still a first messenger. But the difference is, if you are a steroid hormone, you are a first messenger all the way through. They go through the bilayer leaflet. They can bind the receptor in the cytosol, and the receptor will translocate. It's called the mobile receptor hypothesis. It will mobilize. So not every single steroid-based receptor is going to bind in the nucleus. It will eventually end up in the nucleus, but some of them, they go all the way and bind receptors that are bound to the chromosome, and others, they take a different route. It's called the mobile receptor hypothesis. So it binds the receptor in the cytoplasm like a transit trip, and then it will mobilize. But in the end, they will all end up here. And they will transcribe for me mRNA. They will transcribe mRNA, and the mRNA, once transcribed, is going to be translated in the ribosome. And once translated, is going to give me my target protein. And my target protein is going to give me my target effect. So here, my first messenger did everything. Didn't need help from nobody. But if you are polar, like epinephrine, now epinephrine is not steroid based. It is polar, and we call them what? What's the name of the class of hormones that are not steroid based? They are known as, begin with a C. C-A-T. C-A-T-E. Catty. Yeah, catecholamines. So these are catecholamines. So catecholamines. 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 These catecholamines, they bind from the outside as first messengers. Then when they bind, the receptor is going to dimerize, and when the receptor dimerizes, it's going to undergo some conformational change, and that will elicit an intercellular signal transduction pathway. And this intercellular signal transduction pathway is going to go and activate adenyl cyclase. Then adenyl cyclase is going to convert ATP into cyclic AMP. That is my second messenger, cyclic AMP. Then cyclic AMP is going to cause the protein kinase to become activated. Once the protein kinase is activated, then it will go and add a phosphate group known as phosphorylation. It will phosphorylate other enzymes, and then these other enzymes who get phosphorylated, they give me my target effect. So in the second alternative pathway, what happened was the target effect was achieved not by the first messenger, but by the second messenger, due to the first messenger epinephrine catecholamine incapacity and inability to penetrate the bilayer leaflet because it's lipophilic and it's hydrophobic. Okay? So that's how we have seen that genetic expression affects my proteins, and my proteins affect my cell division. Is the cell going to divide? When will the cell divide? In growth and development. Going from being a toddler, an infant, a toddler, then moving into early childhood, then adolescence, then early adulthood. That requires a lot of growth and development with a lot of cell division. Healing, when there's some sort of, when there is some sort of stress like a laceration or an abrasion or some form of corrosive agent that injured my epithelial tissue. Okay, so we begin with a stress. This stress could be a physical agent. The physical agent is an example of that is an increase in temperature, so hot. Increase in temperature or a decrease in temperature, so cold, that will cause damage to the integrity of your tissues. Or a chemical agent, what's an example of a chemical agent? That would be a corrosive agent, and that corrosive agent is going to be like bleach for example, or detergent. I'm sorry?
I'm just saying Clorox.
Yeah, Clorox and all of that, burning the integrity of the tensile tissue of your skin. And also ultraviolet radiation, and trauma like a laceration or a cut, or a bruise when you rupture, like you punch some form of blood vessel and all the blood rushes out, leading to that redness. So all of this stress to your tissue has the ability to fight back. But if these factors, if the intensity is so severe, the intensity, meaning so hot or so cold, chemical agent is so corrosive, the trauma is so severe, ultraviolet radiation is so high, if the intensity is high, or if the duration is long, the duration of the stress is long, and if the onset was so rapid, it happened so quick, the stress was like a knockout punch, it happened so rapidly, so quickly, then the stress is going to develop and lead to cell injury. So the cell is now injured. If the cell is injured, then your mitochondria is going to assess, should I do apoptosis? Should I kill the cell through programmed cell death and remove this dead tissue so that I can begin the path for cell division and healing? Should I begin the healing, or should I give the cell some chance to repair? And that depends. For example, misfolded proteins, that's not a stress. You know, Alzheimer's disease, one of the main causes for Alzheimer's disease is the cell is supposed to commit apoptosis, but is not. So you have so many defected cells that are supposed to undergo apoptosis, and new cells come in their place, but they're not dying. So they keep piling up in the brain, and the patient gets Alzheimer's disease. Mad cow disease, Creutzfeldt disease, very similar. This happened to human beings, that's happened to cows, the same, almost the same. And what happens is cocaine addiction increases the rate of misfolded proteins. So these misfolded proteins pile up on the brain tissue until they damage the brain tissue. Those proteins, when they misfolded, there's something supposed to be misfolded protein response. So when there's an alarm that was supposed to go on, happen in the cell. Hey, this is a tertiary protein that's not folding properly. It's going to now become a stress. It's supposed to fold properly to help me, but it's now going to become a source of damage. And it's now going to become a source of stress for me. So the apoptosis, there are systems that recycle. And remember the proteasome. The proteasome is going to shred. It's a shredder of the proteins. It's going to shred these dead proteins. But if there's so many of them, the transport capacity maximum, they have all been saturated. So now you have a turnover rate, a very high turnover rate, and that leads to stress. So this misfolded proteins, and then the mitochondria is now going to have to start apoptosis. And apoptosis is to program the death of the cell. So the cell has realized that it is damaged, so cell injury now moves on to cell death. So the cell has to die. Why? Because the cell is damaged beyond salvage. Damage beyond salvage or damage beyond repair. So now there's no hope. So we need to remove it. But we have to be very careful. It's like a nuclear plant. You got to be careful when you remove it, because if it breaks, if that cell membrane breaks, all the lysosomes get out of the tissue. All the hydrolytic enzymes will be spilled out in the tissue. All of the metalloproteinases, all of the degradative enzyme is going to degrade my tissue. So I have to convert it from a cell into edible blebs, from a big cell into small, harmless, edible blebs. These harmless edible blebs, your body can dispose of them easily. But the problem is when the damage is not, this is through apoptosis. Apoptosis or cell suicide or programmed cell death will give us harmless edible blebs. But necrosis, what happens in necrosis is that the cell membrane would rupture and all the lysozymes, the degradative enzymes, the proteolytic enzymes, okay, the metalloproteinases, the reactive oxidative species, they're all going to be spilled out in the tissue, damaging nearby tissue. So that's now going to be a problem, that necrosis. But in order to protect ourselves from the necrosis, the mitochondria is going to initiate programmed cell death. But this programmed cell death is a cascade, is a pathway, is mediated by proteins known as caspases. When you hear caspases, that's like a domino effect. So this caspase is like coagulation, like the hemostatic platelet plug formation, like making a thrombus. So there is a cascade, a positive feedback. So each caspase participates in the cascade of initiating apoptosis. But to do that, apoptosis is expensive. Necrosis is cheap. Necrosis is damage, obliteration and incineration of the cell. Everything is spilled out. That's cheap. That doesn't need energy. It is the regulated process of programmed cell death. That's the one that requires a lot of ATP and a lot of oxygen. That's why it is not a secret anymore that hypoxia is necrosis. Because there is not enough oxygen. Like patients who have heart attack, patients who have a stroke. What is a stroke? What is a heart attack? Heart attack is dead heart tissue. Stroke is dead brain tissue. Dead through what? Apoptosis? Through necrosis. Because there is not enough oxygen perfusion. There is hypoxemia. Not enough. The blood tissue, the heart tissue is not receiving enough blood due to the atherosclerosis, due to the plaques, atheromatic plaques, due to the lipids clogging the blood vessels. Not enough oxygen is arriving to the blood, to the heart tissue. So there is a drop in oxygen. That's a form of a stress, hypoxemia. Hypoxemia leads to cell injury. Then it exacerbates cell intensity increase, duration increase, onset increase. That leads to cell death, tissue death, damage beyond salvage. The mitochondria will say, I need oxygen to start apoptosis. Because if we had oxygen, we wouldn't need you to start apoptosis. So now the whole cell is going to be damaged with necrosis. That's heart attack. The same thing happened in the brain. The same thing, just different tissue. No oxygen. The mitochondria doesn't have oxygen, fuel, oxidative phosphorylation, aerobic respiration to initiate this very important apoptotic pathway. So an example of what is the importance of apoptosis is that if you don't initiate apoptosis, you're gonna have a pileup of damage beyond salvage cells. And they're gonna pile up on each other until they make very stressful environment that leads to diseases like Creutzfeldt, also known as mad cow disease, also known as Alzheimer's disease. So that's a form of stress. So you need to appreciate that this tissue needs to regulate cell division and cell death. So going back into any questions here? Yes.
So like the heart attack example that you gave, right? So if, let's say the damage just started and then the mitochondria needs like more energy. So like pulling all that oxygen from the body to do that, that's like gonna increase the stress of the body.
Of course. But it's going to need, it doesn't need energy. It needs oxygen. It needs oxygen to make energy. We're gonna learn later on when we do metabolism that oxygen is the final electron acceptor in the electron transport chain within the mitochondria, and that's how we're gonna get the ATP. So the mitochondria gives ATP, but it doesn't need ATP. It needs oxygen and gives us ATP. So like you said, it's a positive, vicious cycle of extra demands for oxygen. So we need oxygen, but oxygen cannot get there because the blood vessel is clogged. So the blood vessel used to give me oxygen. Now there is an atheromatic plaque. Only a little bit blood is passing through. So the tissue is not receiving enough blood, not receiving enough oxygen.
Okay, so what I'm getting to is, if that's happening and the mitochondria is demanding more oxygen, isn't that gonna increase the damage?
Yeah, it's going to increase the damage, and the cell is going to die by necrosis. It's not gonna wait for the mitochondria. It's gonna rupture, the cell membrane is gonna rupture, and all the material is gonna leak out. And that's gonna lead to inflammation. So necrosis, that's the opposite. What is the opposite of apoptosis? That's necrosis. What's happening? What is the opposite of apoptosis? That's necrosis. Necrosis is vicious damage, rupture of the cell membrane, leakage of all these dangerous lysozymes that destroy nearby tissue.
So it's not just death.
Yeah, it's death in a very vicious way that harms the neighbors. You know, the tissue is like you guys sitting with each other, but there's no space between you. You are attached. Remember we talked about the cadherin and the CAMs? Okay, cell adhesion molecules, they connect them, they are stuck to each other. So if one spills out any of its lysosomal degradative nuclear plant, is gonna destroy nearby tissue.
Around the tissue.
Yeah, and that's gonna lead to inflammation, acute inflammation. That's the first phase of inflammation, where the blood is going to arrive to the tissue, bringing white blood cells. So now the immune system is going to come in to try to carve out all of this necrotic debris, all of the dead tissue. So these macrophages are going to arrive. These neutrophils are going to arrive. And don't forget that another form of stress is the infectious microbes. These infectious microbes cause stress that damage cellular tissue. So the immune system is going to fight these microbial infectious organisms with eosinophils, with basophils, with neutrophils, and then remove all the necrotic tissue and debris, and have a clean space so that healing can take place and regeneration. If the tissue is regenerative, not every single tissue out there has the ability to regenerate. Your skin is regenerative. It can increase the rate of its cell division and make up for cells that are lost. The bone tissue is regenerative, excellently regenerative, but nerve tissue is not regenerative. Nerve tissue is not regenerative. So that is known as tissue in the state of quiescence, G0 phase. We will talk about that, G0 phase quiescence. While the other, that is terminally dividing tissue. It is terminally locked in a state of quiescence. It's quiet forever. It grows one time, it stays there. So you got to be taking care of it and not damaging it, because once it's gone forever. But some other tissues, they can regenerate.
Like a tooth enamel,
I'm sorry?
like a tooth enamel, once the enamel is gone, it's
Yeah,
gone forever.
yeah, it's gone forever. Yeah, so that's bone is a form of a bone tissue. That's a form of cells, osteocytes that are, we're gonna talk about them. Osteocytes give me bone tissue, bone matrix. And we're going to have acute inflammation, then chronic inflammation, and when inflammation is done, we're going to have healing under the condition that the tissue is regenerative. If the tissue is not regenerative, it doesn't divide, okay? It's terminally locked in a state of quiescence. It will never divide again. How is it going to heal?
You have to remove it.
I'm sorry?
I read something.
Huh?
I read something the other day. Like
What is it going to heal by?
neuroplasticity.
So a debridement, like to remove this issue.
Yeah, so the debridement happened with inflammation. The inflammation carved out all of the tissue, and all of the immune cells removed. They removed all the microbes. Now we need to move to the next phase, healing. No more fight, no more inflammation. Now we need healing. But the tissue is not dividing. It's not the skin tissue, it's not the bone tissue. It's some other form of really sensitive tissue that is terminally dividing. But it still needs to heal. How is it going to heal?
Protein, vitamin K.
How are you going to heal with protein? You are a cell. You are a cell who died. So you need cell division. You need more cells. But these cells are...
Stress.
So it is protein actually. What kind of protein? It's like cement. You're gonna dump in cement there. Yeah.
So,
Fibrosis, fiber tissue, fibrosis. You're gonna put collagen, you're gonna put fibers in there, and you're gonna put in it. So we have no cells, but we just dump cement in there. We're gonna dump fiber. That's called healing by fibrosis or scar tissue formation. Scar tissue formation. Scar tissue formation. Or healing by fibrosis. So specialized cells known as fibroblasts. Fibroblasts. Blast meaning build up. We're going to talk about osteoblast. They build up bone tissue. Osteoblast build up bone tissue. And fibroblast build up and make fiber tissue. So if the tissue is not regenerative, we heal by scar tissue formation, and fibroblasts will come and they will dump fiber, healing by fibrosis. Okay, so now I think we should at this point appreciate that the cell must have the capacity through genetic expression by going inside the nucleus and being able to regulate the process of by turning on enzymes on and off through phosphorylation with protein kinases. We need to now talk more about genetic expression. So how does genetic expression take place? So you know that we have to go inside the nucleus now. We have to go inside the nucleus, and inside the nucleus we have our DNA. Inside the nucleus we have our chromosomes. And we have about 46 pair of chromosomes like that, and they are joined together by a centromere into 23 pair. So you have 23 of those X's and you have 46 of those singles. Okay? You have 23 pairs or 46 sister chromatids.
okay, so it's either or. It's not.
It depends on the cell, but that's how they exist. They either exist as a pair in the shape of an X, or they can exist singularly at 46. So 46 hot dogs or 23 X's. So that depends on the stage of the cell. In the end, this is my DNA. When they took out the nucleus from a heart cell of the frog, so you have a skeletal muscle cell, skeletal muscle cell of the frog, and you have the ovum, and that is the sex cell of the frog. They removed the nucleus of the ovum, so now the ovum has no nucleus, and they removed the nucleus of the skeletal muscle cell of the frog, and they put it inside in place of the nucleus of the ovum. What happened? No, it grew perfectly. When a sperm came, it fertilized that ovum, and the ovum was fertilized, and the process of gastrulation took place. And you have morula followed by blastula, and the cells go from 2 cells into multiple cells that give you a tissue. So what did this experiment elucidate? This experiment showed that it doesn't matter what cell are you looking at. You have the same DNA. But this same DNA is useless. It is the machinery inside the cell that makes the DNA do what the cell wants. So this ovum has ovum proteins that ensure that the cell grows histologically, structurally, to become an ovum. But the nucleus is like you have $100 and somebody stole the $100 from you. With the $100 you buy milk and honey. That person with the $100 is gonna go buy beer and something. It is you who gives the $100 its identity. Same thing with the DNA. The DNA is found on each cell, but each different cell, the nerve cell, the heart cell, the smooth muscle cell, the neuron, the hepatocyte, they will use a certain part of the DNA to give them their nature. So, again, if you have $1,000, somebody who is an athlete took that $1,000 from you, they're going to go buy sneakers and they're going to go buy stuff for their life. If a cop took that $1,000, they're going to go, for example, buy a gun, buy whatever for their job. If a chef took that $1,000, they're going to go buy the cooking utensils. You know what I mean? It's not the $1,000. It is who gets that $1,000. They use it in a way to, structurally and functionally to, can you say it again, Candace?
Beneficial for them.
Beneficial for them, exactly. So when we took the nucleus from the skeletal muscle cell of the frog, if you kept it there, that skeletal muscle is going to have a lot of cytoskeletal proteins. Cytoskeletal proteins. It's going to have a lot of cytoskeletal proteins because the muscle needs a lot of cytoskeletal activity. But the ovum doesn't need that. The ovum needs cell division. So when you take the nucleus, you take the $1,000 from a chef, you give it to an athlete. The athlete is not going to go buy sneakers and basketball and tracksuit. He's going to buy the utensils if he's a chef. So now the nucleus that was taken from the muscle cell, put in the ovum was able to, because it has a set of proteins and polymerases that will target certain regions. So in the DNA, this DNA is really long and it's for everybody. If you are a skeletal muscle, you have polymerases that will target only this region. They will transcribe only this region, and then that will get translated, and when it gets translated, it will give you your proteins of interest. But if you, that's for skeletal muscle. Skeletal muscle. But if you are an ovum, you're gonna transcribe only this region. So the polymerase of the ovum are gonna transcribe this region to give it all the proteins it needs to divide from ovum, gastrulation, to morula, to blastula, and so on. And then to give me an organism. So the nucleus is not the issue. The DNA is not the issue. It is the set of polymerases. This team of polymerase this gang that work together to polymerize for me the mRNA that will later on be translated to give me some form of a terminal protein.
So transcribe translates a protein of independence of what?
Yeah, so can you say that again?
Protein of independence, interest.
Protein of interest. Yeah, what I need. I'm a chef. I need cooking utensils. You give me $1,000, I'm gonna go buy cooking utensils. I'm not gonna go buy other stuff. I'm an athlete. I'm gonna go buy sneakers. I'm gonna go buy tracksuit. I'm gonna go get a gym membership. I'm a police officer. I'm gonna go buy a gun. So the $1,000 is gonna be spent differently depending on my structure and my function.
Shouldn't they give you the gun for the gun?
I'm sorry?
Shouldn't they give them the gun for the gun?
But I'm just using that as an example. Just using it as an example.
So it's part of the uniform.
Okay, so here to show you that this DNA is like a reservoir. It is for everybody. It is your proteins as a cell that will target and will make the experience of transcription different based on what you need as a cell. Structurally, you look different from other cells. Functionally, you have a different job because the subset of team of polymerases that work to give you the target. Because if you transcribe this region, you're gonna get a certain protein. Transcribe this region, you're gonna get a certain protein. Transcribe this region, you're gonna get a certain protein. Is that concept clear? This concept is very important. So that's how genetic suppression works. By turning off and on certain regions when I'm on demand. I demand proteins, then I turn on transcription. I don't need any more. I turn off transcription. I'm gonna post this lecture up so you don't need to take photos. If you want, go ahead, but I will post it up for you. Okay? These lectures are all on my YouTube. Maybe I didn't make an announcement. Okay, so if it's gonna be there. You like double job? You like to? So you're gonna see it there. Don't worry about it. Okay, so now just focus.
Ovum protein polymerase.
That's an example. Don't worry about the ovum. Just worry about the concept.
Okay.
That it is the DNA the same on every single cell, but it does not mean all of it is transcribed. It is my set of polymerases who will go. And you guys don't remember polymerases. So now I'm gonna go over polymerases. So polymerases, what are they? They are enzymes. What are enzymes? They are proteins. So what do polymerases do? What do polymerases do? Catalyze proteins. Polymerases, they catalyze proteins. So polymerases,
So break down.
Polymerase. Whenever you see an ase, catalase, peroxidase, polymerase. ACE is an enzyme. ACE is an enzyme. Polymerase makes polymers. McLeod.
Yeah.
Polymerase makes polymers. So if I have a polymerase, I'm gonna end up with polymers, a really long polymer. I'm gonna polymerize, make a polymer, because we said monomers are small, so I'm gonna go from monomer into a polymer.
So poly is more.
Yeah, so and he makes more exactly, more. How many kinds of polymerases do we have? Melissa Mann. You don't know. Well, you don't know how many kinds of polymerases we have. Jurist. We didn't talk about the.
I know it's for RNA.
Yeah. So RNA polymerase makes a polymer of
Nucleotide.
RNA.
a polymers of RNA. So I make polymer, yeah, it's nucleotide, but a polymer of RNA nucleotides. So I make polymers. So that means RNA is not a small guy. RNA is not a monomer.
Right.
Because you don't make monomers with a polymerase.
Right.
You make polymers with a polymerase. So mRNA is big. Is it as big as DNA? Of course not, but it's still big. Okay, I'm not big as Shaq, but I'm still big. So RNA is big, but it's not as big as the DNA, but it's a polymer. It's not an infant, it's a polymer. Who makes the RNA polymerase? It's who makes the RNA? It's an RNA polymerase. But let's say I make a polymer of DNA. I make a polymer of DNA. If I make a polymer of DNA, am I going to use an RNA polymerase? What am I going to use? You're going to use DNA polymerase. DNA polymerase is going to give me the DNA. So now it is no longer a matter of dispute that when I say DNA polymerase, I'm going to make a polymer of DNA. I'm going to make a DNA polymer. And if I'm using RNA polymerase, I'm going to make an RNA polymer. The problem now is, who am I dependent on? What do I depend on to make my...
Those two are the types that you're asking.
Yeah. DNA polymerase and RNA polymerase. And don't forget your central dogma of biology. Central dogma of biology says we go from DNA, and that's a nucleotide
And then mRNA.
polymer. That's a nucleotide polymer. And we make mRNA. We make mRNA. And that is also a nucleotide polymer. He's not a small guy. And then we translate and we get proteins. These proteins are structural, such as collagen. Where do we find collagen? Bone and ligaments. And keratin. Where do we find keratin?
Nails.
Hair and nails. That's structural. Or functional protein such as hemoglobin that transport oxygen, or myoglobin that store oxygen. If they ever ask you what's the difference between hemoglobin and myoglobin, myoglobin store oxygen. It's just like a warehouse. But hemoglobin deliver oxygen. So if hemoglobin and myoglobin are working together and both of them have oxygen, and they arrive at a tissue that need oxygen, who's going to give the tissue oxygen? Hemoglobin or myoglobin? Hemoglobin. Myoglobin will never give tissue because it's just there for storage. But in the end, they are functional. They are not structural. They are not collagen. They are not keratin. And we get them through translation. Where does translation happen? In the cytoplasm. What is the cytoplasm made up of? The cytosol. That's the liquid colloid jelly, white color, because it has a lot of proteins. And then you have the organelles. One of the organelles is the organelle of interest, the ribosome. Translation. And that is not a nucleotide. That is an amino acid.
So polymer, amino acid.
Yeah, and that's also a polymer. So the protein is also a polymer, but it's a polymer of amino acid. Excellent. That's a polymer of amino acid. That's a polymer of nucleotide. And we don't have to say polymer of nucleotide, we can say nucleic acid.
Okay.
Nucleic acid. That means a polymer of nucleotide. That is a nucleic acid also. Nucleic acid. That's a polymer of nucleotide. And this is a protein, a polymer of amino acid, that's a protein. And this process is known as transcription. Transcription. Where does transcription happen? In the nucleus, inside the nucleus. All that you have to know.
Right.
And the DNA polymerase. So now, going from DNA, so this is very important. Going from DNA to mRNA, going from DNA to mRNA, which kind of polymerase do I use? McLeod. You see, this is not trick question. This is very simple. I'm going from DNA to mRNA, who is a nucleotide polymer. Which of these two polymerase? There is no third one.
RNA.
Excellent. Because I am making a polymer of RNA, so I use this one. RNA polymerase. Who did I depend on when I made my mRNA? Who did I read it off? Who did I transcribe?
DNA.
DNA. So it's a DNA dependent RNA polymerase.
Okay.
Here, this going from here to here, that's a DNA dependent RNA polymerase. Okay? DNA dependent RNA polymerase. Now, when I have reverse transcriptase, so reverse transcriptase,
So like HIV.
excellent, is for the retrovirus HIV. What it does is it goes from mRNA nucleotide polymer into DNA nucleotide polymer. So reverse transcriptase is what kind of polymerase? DNA polymerase, because it's making for me a DNA polymer. So it's a DNA polymerase. What did it depend on to give me the DNA polymer?
RNA.
RNA, depended on RNA to give me, so it has, the virus has on its capsule RNA. It will take that RNA and it will get it translated, it will get it transcribed because it has the enzyme, reverse transcriptase. It will get it transcribed in a reverse manner to give you DNA. And that DNA is going to be later on transcribed with, like in your cell, in your body. The virus has no organelles. The virus just has the mRNA transcript. It has also that enzyme, reverse transcriptase. So when it infects your cell, it will initiate reverse transcriptase to do a RNA dependent DNA polymerase and make a DNA double strand.
Right, because the mRNA is gonna hijack the DNA.
It's not the mRNA. Your DNA will get completely inactivated. Your nucleus DNA is destroyed. Now it is the virus who's going to do reverse transcriptase its own mRNA to a viral DNA.
Okay.
Then it will use your ribosome to transcribe its DNA. It will use your polymerases to transcribe its DNA, your ribosome to translate that mRNA.
Okay.
That's how the virus works. So this is an example of the retrovirus. The retrovirus is going to give me DNA. So it's a reverse transcriptase because transcription is in this direction.
Yeah.
So that's why they call it reverse transcriptase. So it's a DNA polymerase dependent on RNA. So it's an RNA dependent. It's an RNA dependent DNA polymerase. The last one happens in replication. If a cell wants to divide in the cell cycle, like in growth and development, as well as in healing, what happens? You have a cell with DNA 1X. Then you're gonna replicate through the process of replication, through replication, we're going to replicate and we're gonna double the DNA, and now the DNA is gonna become 2X. The DNA is gonna become 2X. Then we're gonna go through mitosis. Mitosis, cell division, all the different stages of mitosis. You have prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis. And then you're going to have two cells that are going to divide as 1x each, and then you're going to have two cells that is 1x each. So this cell division happened because I doubled the DNA. If I want to double this DNA, obviously I doubled the DNA. So what kind of polymer am I going to use if I double the DNA? What kind of polymer?
DNA polymer.
That's excellent. That's a DNA polymerase. But what did I read off of? I used to look like that. That's my DNA. Used to look like this. Double-stranded DNA is a double-stranded DNA, double helix. Then I'm going to separate it and replicate it, and I'm going to make a copy. I'm going to make a copy of each. I'm going to make a copy of each daughter strand, the leading strand and the lagging strand, and then I'm going to have copied my DNA. So I doubled. You see the red one wasn't there. I cut it, then I doubled it, and now I have it. So who did I depend on? Did I depend on RNA? I depend on DNA. So it's going to be DNA dependent DNA polymerase. DNA dependent DNA polymerase. So that's for replication. So you need to know all three. DNA dependent RNA polymerase. RNA dependent DNA polymerase. DNA dependent DNA polymerase. That's for replication. So now we got to talk about replication. What time it is? 1 more hour. 1 more hour. God will pay me in the day of judgment. Okay. I hope I go to heaven. Anyways, let's now go through. Let's go through the replication. That's very, very important. The replication, really important.
This one is really.
Okay. All right. So now replication. The DNA, we said, the DNA must have nucleotides that are either adenine, thymine, guanine, cytosine, guanine, thymine, guanine, adenine, thymine. And who pairs? Who pairs with adenine? It's always thymine. Who pairs with thymine? Adenine. Who pairs with guanine? Cytosine. guanine, cytosine, adenine, cytosine. That's the easiest thing. I think there's a question on the exam about that. Very simple.
Thank you.
You're gonna get like one strand, and then you have to
Can you put like 6 of them up there?
yeah, something like that, and then you have to like just get the opposite. Anyways, if I want to divide, so I have to double everything up. What happens is the DNA polymerase is gonna come in. It's a set. It's a whole set of team that work together. They're going to first of all helicase. Helicase is gonna come. Helicase will come, and it will unwind for me the strand. It's going to unwind the strand for me. Helicase.
Helicase. Okay.
It's going to unwind the helix. That's why they call it helicase. It's going to unwind the helix. After it unwinds the helix, you have two strands. You have the leading strand, and you have the lagging strand. Why is that? Why do you have a leading strand and why do you have a lagging strand? Because the DNA is anti-parallel. The DNA is anti-parallel. Like two snakes. One with head up and tail down. The other one with head down and tail up. One is 5' to 3'. The other one is 5' here to 3'. They are anti-parallel. So for me, DNA replication, I needed number one, helicase came in, it unwinded the DNA. Number two, my DNA polymerase is going to come and bind. DNA polymerase is going to come and is going to bind my leading strand. And my leading strand is going to be from the 5' to the 3'. So the DNA replication is going to be from 5' to 3' for every AT, for every TA. But the problem is, the replication always has to be 5' to 3'. But the problem is, the other strand is anti-parallel. So now you have 5' here to 3' here. And the transcription is happening in this direction. Transcription is always happening from 5' to 3'. So that means on the opposite direction, you're going to make 5' to 3' reading, but it's going to be discontinuous. Discontinuous. It's going to be discontinuous because now we are going to be discontinuous because we are going in the opposite direction. And then there will be fragments that are going to be staying behind, lagging behind. Those have to be joined, glued together by an enzyme known as DNA ligase. It will glue together these fragments, these chopped up, these cut up fragments. These chopped up and cut up fragments are known as Okazaki fragments, named after the scientist who discovered them from Japan, Okazaki fragments. These Okazaki fragments are going to be joined together, and in the end, I'm going to have one whole strand. So then I'm going to separate, and I'm going to look like that. That's my lagging strand. And that is my daughter. This is the daughter. This is the parent, the original strand, or the parental strands. So I have this is the first parental strand, and it's lagging because it's cut up, discontinuous. And then there is another parental strand, but it's the leading because it's in the right direction. So there is a leading parental strand. And then there is a lagging parental strand. Like that. Okay? And the two greens are the daughter cells. These are the daughter strands. So now I have doubled my DNA, and now I am ready for cell. I'm ready for cell division. Then the cell is going to divide. The cell is going to divide. Okay, that's as far as replication. So the DNA replication is discontinuous and anti-parallel, and these ligases are going to connect together. I think there's a question on the exam about that. What's DNA ligase? It's going to connect all the strands.
Wait, what?
DNA ligase.
What is DNA ligase?
Connects all.
It connects the cut up Okazaki fragments in the discontinuous replication process. Okay? Whew. All right. Now, before we get out of here, let's now talk about transcription, because this is what makes the cell a cell. That's why when they took the nucleus from the frog skeletal muscle cell and they put it inside the nucleus of an ovum, we ended up with an ovum.
So with the ovum,
You ended up with ovum material.
right, so, the gastrulation.
Yeah, the process of gastrulation, meaning dividing after the fertilization. So they're gonna divide into cells. They're gonna blast into cells that will give me tissue. They're gonna build up into tissue. Now we're back to the most important thing, transcription. Who are you gonna transcribe is what's gonna give you your identity. The set, it is the RNA polymerase who makes the cell what it is. It's not the DNA polymerase. The DNA polymerase is important for providing. DNA is like the bank. The money's there. But how you spend it is what makes you who you are. So the DNA is there in the nucleus. But how you transcribe it, which region you decide to go to and make proteins, that is what makes you what kind of cell. Okay? All right, so in transcription, what happens is we have DNA. That's DNA. DNA. And in transcription, you're going to get a copy of mRNA. And you're going to get for every A, T, T, G, C, A, G, T, C, T, A, T. Okay, for every A, RNA does not have thymine. It has, thank you, A, C, G, T, T, C, A, G.
So you messed up already.
How's that?
The T is supposed to be a U.
Oh, excellent. You see?
Nice. Look at that.
That's very good. U, A. All right. Now, this is my mRNA transcript, but the DNA is so long. It's so long, but we're not transcribing every region. This is the region known as the operon. That's what I need, the operon. The operon is the area I'm gonna target to make. So my operon as a skeletal muscle cell is gonna be different from my operon as a cardiac muscle cell, is going to be different from my operon as a neuron. So it's the region I'm going to transcribe. That's my operon. Okay. Then I'm going to get my mRNA. So that happened in the nucleus, inside the nucleus. And we said the DNA is so compacted. It's so tightly coiled. It's so tightly coiled because it is negatively charged due to the phosphate group in the phosphodiester bond. In the phosphodiester bond, and due to the phosphate group in the phosphodiester bond, it's gonna bind to the histone complex. It's gonna bind to the histone complex so tightly, and it's gonna get coiled, and the histone is positively charged. And this is negative, so now they're gonna stick together really much. So if I need to transcribe a region, there are proteins and enzymes known as topoisomerase. These, they will create nicks, K-N-I-C-K-S. They nicks or cuts. They will cut for me and make accessible there, because it is so tightly coiled, you cannot really access it. But what happens is you will have acetylases. So these are a set of proteins known as acetylases, and the acetylases, they are positively charged, and they will come in between the negatively charged DNA and the positively charged histone. Acetylases will come and they will repel the histone from the DNA, and then they will cause a gap. That gap will make access so you can go inside and transcribe. And the topoisomerases will cut up the DNA and make accessible that region. So they help the process of transcription. They help the process of transcription. Then, but you don't have to worry about that. Just learn it in a way that makes you appreciate it. But this is not something that you just need to memorize. This is something you need to know, that the process of DNA is so coiled. We need to uncoil it by using these charges to repel the histone from its DNA, and then we're going to use topoisomerases to chop and cut up regions to make them accessible for us to transcribe. Then the mRNA, we're going to have a premature, nascent mRNA. Premature nascent mRNA. This nascent mRNA will have to undergo post-transcriptional modifications. I have to modify. I have to perform post-transcriptional modifications. What are the post-transcriptional modifications that we talked about last week? What are the post-transcriptional modifications that we talked about last week? Excellent, Joris, excellent. So you have, this is an exon. Exon 1, exon mobile, exon 2, exon 3, exon 4. Okay. This is the intron. The problem, students think intron stay in and it's gonna be in the, no, intron is gonna get excised. So we're gonna excise, cut out, exit the intron. So the introns are gonna get removed out, and exon 1, exon 2, exon 3, and exon 4 are all going to be connected. And now that's my final mRNA transcript. And that's going to exit. The nucleus. And it's going to head to the cytoplasm. It's going to exit the nucleus and it's going to head to the cytoplasm for translation in the ribosome. But the introns are gonna stay in the the nucleus. Because they cannot be part of my last transcript.
So the introns is gonna be cut out and exit?
Nope. The introns are gonna stay in the nucleus. See, I put in nucleus. So you see the nucleus, the transcription happened in the nucleus. But translation must happen in the cytoplasm.
Okay.
So the exons exit to the cytoplasm for translation. But the introns stay in the nucleus. They are sequestered, they cannot get out. But we have alternative splicing. And alternative splicing means we cut up and reassemble these introns and recycle them so that later on we can transcribe them in a beneficial way. Questions? This is very important.
I'm still confused.
Yeah.
Because you said the enzyme?
This is my mRNA.
Right.
This is my nascent.
But the purple you removed.
The purple I removed, that's my intron. Don't say the purple. That's my intron. That's the intron.
Okay,
That's the intron. And the red is the exon.
Okay, so the intron it stayed in the nucleus.
We cut it out and stayed behind in the nucleus.
Right, and then the
Red-
exons went up to the for translation into the cytoplasm.
Yes.
Okay.
Excellent. And then we're not done. The introns, we don't let them pile up. They're gonna give us Alzheimer's disease. They're gonna give us the misfolded, and they're gonna give us like so much dumping.
So how do we get rid of it?
So we do alternative splicing. So we're gonna reassemble them and chop them up and recycle them to convert them to exons that can exit the nucleus and be translated to give me some form of useful mRNA that I could use as a cell. Okay? Yeah, we're gonna reassemble them.
Okay.
They are, remember, they are still mRNA,
Yes.
but we didn't need it with the exon. But it can stay behind. Okay? If you're not good enough to represent USA in the Olympic basketball team, it doesn't mean you cannot be used to put some other team in the home, right?
Right.
So that's what they're gonna do. The introns, they're not gonna be part of the exon that's gonna go out into the
God bless you.
ribosome for translation. But it's gonna stay behind, but it's gonna be assembled and they're gonna be put together to make some useful form of mRNA that will exit later on to go play in like somewhere in like some China or somewhere. Okay?
Okay, so like overseas basketball.
Yeah, something like that, yeah. So but it's not gonna be like the exon, the main transcript. Okay? They get spliced out.
Not the NBA, you just went overseas.
Yeah.
Okay.
No.
Yes, that is it.
No, it's not.
It is. If you know what she means, that is what she means. Yeah.
No, she said the NBA.
It's like
We want,
the
hold on.
Exxon is going to the NBA.
No, the exon is going to the Olympics. The
Exactly. It's the same thing.
It's
the
not the
NBA
same.
is the Olympics.
It's
So
the-
that's
No,
what
it's
I'm
not.
saying.
You
No,
know what
it's
I'm
not.
Like, it's
Hold on.
the-
Calm down. Hold on.
Bro, that's the analogy. It's
Hold on.
correct.
Hold on. If you are going to the Olympics, you represent
the USA.
USA. So you're going to represent the USA in
The world.
somewhere.
And Paris.
But NBA, that's a local league for home.
Right.
Okay, so just because you made it home does not mean you're gonna make it out to the Olympics. You understand?
Right.
So the NBA players, they are introns. It is the ones who make it to the Olympics. These are the exons.
Okay.
So if you go out the nucleus to get translated, you are Olympian. But if you stay behind for alternative splicing, you're just going to be recycled for something.
Are you just
Okay.
a player on the team?
So we're now going to move on to. So we have finished transcription. Now we have to go to translation. So now I have my final mRNA transcript. I now have my final mRNA transcript. What am I going to do with my final mRNA transcript? Now I don't need to excise it. I don't need introns. I don't need exons. I need to be translated.
So it's going to translate to its proper.
It's going to translate to its.
The proper job that it needs to do.
It's going to get translated to a protein.
Right.
So what happens now is you have to now learn the different types of RNAs. We have a clover leaf. We have a clover leaf.
That's a cool. You could have said 4 clovers.
That is, we have a clover leaf that has 3 sides. One side is going to have my anticodon, and the rest they just have some complementary codons, but the anticodon is going to be right in the middle, and on the tail you're going to have the amino acid, AA, the amino acid. And here you're going to have the anticodon. What's going on? Are you guys paying attention?
Yeah.
So this is my mRNA. This is my mRNA. That's my final transcript. That's my final transcript. AUG G CU AU AG CG AA. For example, let's say that's my mRNA. My mRNA, how is it going to get translated? The first step is that the small ribosomal subunit, small ribosomal subunit is going to bind in the bottom, and it's going to attach to a ladder sequence. Is going to bind to a ladder sequence. Then the large subunit is going to bind at the top. And the large subunit is going to have multiple sites. It's going to have an entry site that begins with an A, then a polypeptide site that begins with a P, and then an exit site that's an E. So what happens now is this tRNA, is going to come and is going to bind the start codon. This start codon is so special, and you need to know it because the start codon AUG is a start codon, and at the same time it codes for what amino acid? Methionine. It's methionine. So this methionine is going to be up at the tail of the tRNA. So in the ribosome, you have rRNA, ribosomal RNA. And with the amino acids, you have the tRNA, that's the transfer. That's a transfer RNA. And you have your mRNA, your messenger RNA. So what happens now is your mRNA, your messenger mRNA, is going to get bounded on its ladder sequence with a small ribosomal subunit, and this small ribosomal subunit is 40S ribosomal subunit, and then the large ribosomal subunit, that's 60S. Then they're gonna bind, and you will have an anticodon A for AUG. That's going to be UAC. Is going to bind the start codon and bringing in its amino acid methionine. Then it's going to shift from the entry site into the polypeptide site. It's going to stop there. It's going to stop in the polypeptide site. Then another transfer RNA is going to come in. It's going to bind with this anticodon, and a shift will happen from the previous tRNA who was waiting at the P site is going to shift from the P site to the entry site, it's amino acid. Then it's gonna move to the exit site and get out. Then a new transfer RNA with its amino acid is gonna come in, and a shift will happen. We're gonna shift and transfer over 2 amino acids from the previous 2 tRNAs, and then the empty ones are gonna keep moving. But I'm gonna show you a video. It's very important to understand it, but it's not gonna happen without a video. So I'll show you the video. It's gonna make you understand it really well. But the shift happens from the polypeptide, P for polypeptide, E for exit, A for entry. When the tRNA comes in, it waits in the P position, then it shifts the amino acid to the incoming tRNA, then it exits, then that tRNA that received the polypeptide chain is going to shift to the P position, and when the next tRNA moves in, it's going to shift from P to A, and that's how we're going to keep doing that until we reach the stop codon. We have 3 stop codons. You go away, you are away, and you are gone. Once we reach those have no tRNA amino acids. They do not code for anything. They just terminate. These are stop sequences. It's not like the start codon is going to have methionine. It's going to code for methionine. Start the mRNA translation and add methionine. These guys, they just terminate. They do not add anything. Okay. So then we translate our mRNA, and we have a sequence, a primary sequence of amino acids, This primary sequence of amino acids like beads connected by a string. That string represents for me the peptide bond. So they're all connected. And this is a primary sequence. The primary sequence, in order for me to go from a primary sequence to a secondary sequence, I fold upon myself to make an alpha helix, and a beta pleated sheet. This beta pleated sheet and the alpha helix have hydrogen bonding, led them to become secondary. Then I can move to tertiary. What's the most famous example of a tertiary protein? That's your myoglobin. Myoglobin is a tertiary protein for oxygen storage. And it goes from hydrogen bonding to myoglobin because of it goes from secondary to tertiary through more extensive hydrogen bonding and hydrophobic effects. Then I go from tertiary to quaternary structure, and that is through all 3. More extensive hydrogen bonding plus hydrophobic effects, and lastly ionic bonding. So what makes the difference between a tertiary degree protein and a quaternary degree protein is the ionic bonding. We introduce now a new form of bonding. So it's not just It's not just hydrophobic effects. It's not just hydrogen bonding. We now introduce ionic bonding. So where does ionic bonding come in play? It's in the quaternary structure. So what did Christian Anfinsen do and get a Nobel Prize? Christian Anfinsen was a very brilliant scientist. He was a biochemist. And what he did was he discovered that obviously when this is Christian Anfinsen. Christian Anfinsen. What Christian Anfinsen did was that, and he got a Nobel Prize for that. What Christian Anfinsen did was that he realized if a protein falls from a primary to a secondary, we said through what? Hydrogen bonding. When you go from secondary to tertiary, that's through what?
Hydrogen bonding.
And hydrophobic effects. And when you go from tertiary to quaternary, that's hydrogen bonding, hydrophobic effects, and you introduce ionic bonding. He realized that if you change the pH and if you change the temperature, you're going to denature the protein from quaternary to tertiary, from tertiary to secondary, from secondary to primary. But he realized if you restore the optima, if you restore the optimal conditions, you restore the pH, the proper pH, you restore the proper temperature, then you will refold right back. Refold right back. So that means my protein was not entirely destroyed. So he discovered that it is the most important one is the primary structure. This is the manuscript for the enzyme, for the protein. Yes, the protein works at a quaternary structure. Yes, the protein works at myoglobin in a tertiary structure, but it is the primary structure that gave it is the manuscript that gave it its prescription, that gives it its structure and function. So if I restore my optimal conditions, I'm going to refold right back up. I haven't lost anything. So the most essential thing Christian Anfinsen discovered, and he got a Nobel Prize for, was that it is the primary sequence that determines the quaternary, and that determines the hydrophobic effects and the ionic bonding, and that what gives the identity to the protein. But obviously, if you denature like crazy, you make the temperature so hot, now you don't just destroy hydrophobic effects and ionic bonding and the hydrogen bonding. Now you're gonna destroy covalent bonds, the phosphodiester bond. You're gonna destroy all of the peptide bonds, and then that's irreversible now. That's necrosis. That's out of line. But he's saying moderately, it is this primary structure. So when we get our translation, we get it in the form of primary, then it folds. And then that information of those amino acids working together, you have also disulfide bridging. Here you have disulfide bridging that helps the process of the refolding. So that is our translation.
Doing the mannequin challenge?
Sorry?
Are you doing the mannequin challenge? I'm stuck there for a minute. What you thinking about?
Okay, guys, let's now move to. So we're now done with the cell. We're now just done. We're completely done with the cell. Now we're gonna move up to the tissue. We're gonna move up to the tissue, and we're gonna realize that we have 4 different types of tissues. So even though the DNA is different, the cells can have different shape, cellular shape, like for example cuboidal cells, like box. Okay, you have a squamous cell that look like a sunny side egg up, the egg sunny side up, sunny side egg
I
up.
like how you explain that.
Okay.
Sunny side up egg.
Sunny side up egg. Thank you. You have a sunny side up egg, and sunny side up egg, sunflower seed. Sunny. Okay, so you have a sunny side up egg, and you have a box-shaped cuboidal cell, and you have a stellate cell, discoidal cell. Those names you have to know that. That's gonna be on the midterm. You know, I'm gonna post for you one slide. It's gonna have the nine different type of cells. The 9 different types of cells. The discoid cell, like red blood cell, stellate cell, like the nervous cell, cuboidal cell, and epithelial cell, so forth. The 9 are gonna be on the midterm. So that's already 9 points for you. All you need to do is just remember them. And they are very easy. Discoid, that's a red blood cell. We talked about the discoid shape, and that is thermodynamically favorable. And a sickle cell does not have that. It looks sickled from the 2 edges. So that's a red blood cell. So you know you have to know that really well. And did you get the announcement of me opening all that recharge kind of thing? So I did what you asked me.
I didn't see
I'm sorry?
I didn't see the recharge.
Okay, you didn't see it, but I did it. So you can believe me, and then you can go check it home. But I did it. Yes.
Just the genetic inheritance. When you click on it, it doesn't let you open it. It just goes back to view.
I don't get it. What are you saying?
One of them won't open. You want me to show you?
No, you're talking about the labs. I'm not talking about the lab. I'm talking about homeworks, assignments, because that's what's going to be on your exam. So, but I heard from them. The problem is, it's only after the due date that it will happen. Only the deadline. So they come out after the deadline. But you have the study guide. That's all you need. But if you want more questions, you can. Yes.
That's not what we're talking about. We're asking you, the lab that we did last week, that you posted, you said you extended the due date to the 13th, so we could go back in and do it because of the glitch.
It still won't open.
But it still will not open because even though you extended the due date, you didn't unlock it from 1 attempt.
Yes.
So we can only view the past results.
You have
Remember
to
when I
add
was having
more.
the issue where I
Okay.
told you?
Did you send me an email?
Yeah.
You sent us an email and we
I
closed
sent
the
you-
browser.
Okay. I'm gonna.
I sent you an email though.
Okay, I'm gonna reopen them for you guys, and I'm gonna make it available for you guys to redo it again. But yeah, this recharge, it was like something that you guys paid for. I didn't know about it, but you guys told me about it, so I called them and they helped me set it up. So you have your exams that are open with detailed solutions, so you can go redo them again. It's gonna help you a lot with your midterm. Yes.
So you're saying if we did the chapters, like now that's, so if we do the chapters, like the chapter 5 and whatever, that's gonna help us with the, whatever the.
Yeah. So if you, it's gonna help you, but make sure you do the study guide first. You do the homeworks, then you can go to do the recharge. Because in the recharge you may learn, I mean, the more knowledge you get, the better.
Yeah.
But for me, my exam is strictly based on my study guide.
You got it.
Okay?
Thank you.
All right. So now we have to appreciate that this tissue is made up of multiple cells. And we have embryological germ layers. So we have the ectoderm that is all the way on the outside. Ectoderm. That's all the way on the outside. In the middle, you have the mesoderm. And the inner core, that is the endoderm. So most of the tissues, they have shared germ layers. That's why they are very similar. And you also have to examine, for example, the different types of tissue. There's epithelial tissue. Connective tissue. Excellent. Muscular tissue. And lastly, you have nervous tissue. Nervous tissue is excitable. And this is, we are getting closer and closer to this action potential. This action potential that's now we're gonna appreciate that this electrolyte influx and efflux will affect the membrane voltage And that membrane voltage change is going to lead to what? Opening of ion channels. So that membrane voltage, we're going to change it with the influx and efflux of our electrolytes. Calcium, influxin, increase the membrane potential. That's known as depolarization. Now we have our own language. We don't say increase membrane potential. We say depolarization. We don't say decrease membrane potential. We say repolarization. So that is how the excitable tissue, like your skeletal muscle cell, your smooth muscle cell, they contract. The smooth muscle cell contract and relax. Your heart, your cardiac myocyte contracts and relaxes. Your skeletal muscles, your biceps, triceps, they contract and they relax, responding to this membrane voltage change. And the membrane voltage will change because the ion channels open, allowing the influx of some electrolytes and the efflux of some other electrolytes. And this is happening in the nervous tissue and the muscular tissue. They are excitable. The epithelial tissue is always lining the outside and inside. So on the outside, in your epidermis, that's epithelial tissue. It does have nerve cells, but it's predominantly epithelial tissue. Inside your hollow organs, that is also epithelial tissue. Okay? This epithelial tissue lines the outside and inside. It is on your respiratory tract, in your alimentary tract, your digestive tract, from your oral cavity, down to the rectal canal. That's all epithelial tissue. And then you have connective tissue. That's the easiest one because it connects these epithelial tissues to the basement membrane. It has an apical side and a basolateral side. And the perfect example of connective tissue is the blood. Blood is a type of connective tissue. One of the questions that are very high yield on the NCLEX or on examinations, no matter what microbiology, anatomy, and physiology, no matter what it is, they will always ask you, what kind of tissue is blood? What kind of tissue is blood? Is it connective tissue? Is it muscular tissue? Is it epithelial tissue? Is it nervous tissue? It is a type of connective tissue. So we're going to be learning today. We're going to be going over the slides, finishing up where the cell cycle. I forgot the cell cycle. Okay, the last thing I did not do before we go into the tissue is the cell cycle. So the cell cycle is very important because that's how we understand how cancer happens. So in the cell cycle, we have, people say the cell cycle's 3 phases. No, cell cycle is 2 phases. Is interphase, there's interphase, and that is comprised of the G1 phase, the S phase, the G2 phase, and then you have your mitosis. And mitosis is divided into prophase, metaphase, anaphase, telophase, and cytokinesis. So this stage, the most important one, is the S phase, where we double the DNA. Where we scan the DNA for any errors, because you don't want to double up a damaged DNA. You don't want to double up a mutated DNA. So the process of DNA replication has high fidelity. So these DNA polymerases and the RNA polymerases, they have high fidelity. What is high fidelity? Meaning it has very low error percentage. So out of making a billion base pairs, it makes sometimes one mistake out of a billion base pairs. So it has really high fidelity. So we scan in the S phase the DNA. So this is the DNA related phase, the S phase. The G1 phase is where we double the organelles and we prepare the cell for the doubling of the DNA. The G2 phase is also a preparatory phase, but now we are finally finalizing the last moments before the cell enters into mitosis. And there is a checkpoint here. If the cell is ready, the DNA is not damaged, it will progress forward and it will go into mitosis. If it's damaged, it will exit out. And this phase that does not have mitosis is known as interphase, and the other phase is known as mitosis. So we're just going to be going into the slides, learning more about the events that happen at every phase, and then we're going to go into mitosis, knowing about the distinct features. The most important phases is that in metaphase, that is where you will have your chromosomes lined up in the middle, and this is known as the metaphase plate. And the second one is anaphase. That's also very important because then you have the actual successful pulling apart. You have the successful pulling apart when they successfully pull in the opposite direction. The microtubules pull the chromosomes, the sister chromatids, in the opposite direction, and they actually successfully separate. They pull in the opposite direction. So just remember, these are the distinctive features of anaphase and metaphase. But before the cell gets to anaphase and metaphase, you had centrioles. The centrioles, they are microtubules. They will start migrating, they will start polarizing, and themselves they get doubled. So you would have two centrioles, and the two centrioles, both of them will polarize to one pole of the dividing cell. Then after that, they will start to generate a star shape. This star shape structure is going to start to pull itself away from the centriole to attach to the chromosome. That's the first phase, moving away from the centriole to the chromosome. The next phase is anaphase, is when they move away from the chromosome toward the centriole, and that is what causes the division. So they have two different movements. One movement, they move away from the centriole to attach themselves to the chromosome. And then they move back towards the centrosome. So this is my centromere. These are my centrosomes in the opposite directions. They have doubled. They have went to opposite directions, opposite poles. And now these very important microtubules move away from the two poles of each of the centromeres, and then after they move away, they attach to the kinetochores. And we said last week that you have the middle of the chromosome known as the centromere, and on it you have a plate known as the kinetochore, and these microtubules gonna move away from the centromere to the kinetochore. That's the first phase. The second phase, they're gonna move now away from the chromosome and back respectively to its centromere, so they can pull off and pluck off these two sister chromatids that are attached. So we will see more figures, and that's gonna help us more. Yes, you have a question? You're scratching your eye. Okay, all right, so let's come back at 11 in the other room. Anybody has another question? Okay, let me take attendance also.